Okay, so we've finished the overview of the structure of RNA versus DNA. So now let's discuss their function, how they are similar and how they are different. DNA functions as a blueprint for the synthesis or the making of proteins, which is very important because proteins are what I like to call the doers and determiners in an organism, because proteins have a lot of roles in the cell. They do a lot of things, and they're largely responsible for determining how we grow, develop, look, and reproduce. DNA, which again are the original instructions for proteins, are found in nearly all living cells. So almost every cell in your body has DNA, and DNA is inherited from the parents to their offspring. RNA is special because it's much smaller than DNA. Its size allows it to travel throughout the cell very easily. For instance, DNA is usually found in an organelle called the nucleus. Now, the nucleus's membrane has tiny microscopic holes in it we call pores. Imagine the pores on your face. They look kind of like tiny holes. Same thing here. So the nuclear membrane has tiny nuclear pores, but DNA is pretty bulky. It is very large, long, double helical in structure. Because of this, it's confined to the nucleus. It's too large to squeeze out through these small holes. But RNA is special. RNA is comparatively quite small and can fit through these nuclear pores and travel in and out of the nucleus, meaning it's capable of carrying instructions or information from the nucleus to almost anywhere in the cell that it needs to go. Because of this, RNA's main role is to act sort of like a messenger, to convey instructions from DNA to other parts of the cell to help make proteins properly. Now, this is just a general overview. RNA actually contributes to the cell in many different ways because of its ability to travel and interact with other molecules. But for the purposes of this introductory lesson, this is what I want you to take away, the big takeaway. This is the big picture you should hold on to for now, and the fun and interesting nitty-gritty details are to come later on in this semester. Sound good? But this information here actually ties in beautifully with what the relationship between DNA and RNA is. DNA, as we went over, is the blueprint for making proteins. And DNA is found in the nucleus of most cells, but the structures that actually do the building of the proteins are outside of the nucleus. Since DNA is too big to leave the nucleus, how do these structures get their instructions for building proteins? By making RNA strands that carry the same information as DNA to other parts of the cell. So how is this done? Well, inside the nucleus, a protein binds to part of the DNA and unwinds it a bit. This protein then builds RNA using DNA as its template. So let's zoom in on this for a closer look at what's happening. So we have our protein here that is bound to DNA, and it is opening a part of the DNA's double helix up to make space for the protein to then make some RNA. Now it makes RNA by finding free nitrogenous bases that complement the nitrogenous bases in the DNA strand and linking them together. Remember which nitrogenous bases pair with which? Well, here's a reminder, just in case. Remembering that uracil is used for RNA. And now this small RNA segment is good to leave the nucleus and transport part of the DNA's instructions to somewhere else in the cell. So now somewhere else in the cell can get at least a part of the DNA's instructions, thanks to RNA. And that's the brief overview of nucleic acids. So, in summary, well, actually, let's have you do the summary as your next check-in question. For this check-in, you're going to make a table evaluating the relationship, the function, and the structure for both RNA and DNA, as well as for just RNA and for just DNA. So in the first column of your table, you should record the similarities in structure between RNA and DNA, the similarities in function between RNA and DNA, and then the bottom row, for the entire row, you can just write the relationship they have to one another. So the answers in the bottom row can all be the same. The second column should go over the structures and functions that are unique to RNA, so RNA only. And the final column should go over the structures and functions that are unique to DNA, so DNA only. To do this, you're going to need to go to Canvas, and you're going to click on this table icon here, and then you're going to make a 4x4 table. And then you're going to fill in the column and row titles like you see on the screen here, and then fill in your table. Obviously, don't write squigglies. And although this is a prompt for a check-in question, I actually highly recommend you write down this table in your notes as well. It'll help you with studying, for sure. But wait, there's more! Now what we just finished up with is an overview of what people generally associate with nucleic acids. However, there is one more nucleotide worth mentioning. Even though it does not necessarily fit in with the discussion of DNA and RNA, it is still a nucleotide, and that nucleotide is ATP. ATP stands for adenosine triphosphate. And it is completely unique and different from other nucleotides because first, it has, well, the same overall base, but 3 phosphate groups. Hence why it's called triphosphate. And second, it does not polymerize, which means it is always a single nucleotide. It never bonds with other nucleotides to form a nucleic acid. It is always a single nucleotide. Something that is also unique to ATP is its function. It does not serve as directions like RNA or DNA at all. ATP is actually known as energy currency. So whenever you see ATP pop up in a subsequent discussion, I want you thinking energy. So if a cell has one ATP molecule present, then it effectively has 1 unit of ATP. Don't let this confuse you though. Generally speaking, if we are talking about nucleic acids, we are talking about RNA or DNA, and we are talking about molecules whose main function is to carry information inside cells. ATP is technically a nucleotide, but it does not polymerize, so it's not really a nucleic acid. It is just a monomer of a nucleic acid, and it will always stay a monomer. And its main quote-unquote function is that it is energy for the cell. So if you hear nucleic acid, you really should be more thinking about this over here, okay? Just nitty-gritty details, ATP is technically a nucleotide.